Hey everyone, welcome back to another video. I noticed today I've been talking a lot about DNA and RNA, but I haven't had the time to properly explain it to you. Today we'll be talking about genetic material, which is basically DNA and RNA. And first of all, let's go into what are they. So, as you can see, this here is an RNA, ribonucleic acid, and it's just one single strand with all these base pairs, which are called nucleobases. And this is a DNA, which is deoxyribonucleic acid. So this is ribonucleic acid, deoxyribonucleic acid. And what separates these two mainly is the structure, because DNA, as you can see, has this like a curve, it's twisted and it's has two two sides. So basically, both of them compose of a backbone, which is a sugar phosphate. But then they also have bases, like base pairs here for DNA and nucleobases for RNA. RNA. So before I go into that, every single living thing uses DNA. Even viruses. Viruses mostly use RNA. Some can use DNA, but every single thing that we can consider living has some form of this. And if you come ranges from bacteria to plants, every single living thing. That's what kind of characterizes. That's one of the characteristics of life. So, as you can see here, we have three things, three bases. Let's go over the DNA first. So, as you can see, it's a helix shape, helical shape, meaning it's like a twisted, curvy, like I know it's not 3D, but it's kind of twisted and 3D curvy, and so basically, you can think of the pairs like ATCG, which is adenine, thymine, cytosine, guanine for DNA. Okay, and A always pairs with T, C always pairs with G, and vice versa. G always pairs with C, A always pairs with T. So using that, you can create multiple number of combinations with this because the DNA can be very, very long. It can be, very long, and you can have as many base pairs as you want. So one base pair consists of either thymine and adenine or cytosine and guanine. And there can be like hundreds of thousands, millions, billions, trillions of these base pairs in like a human genome. Recently, us humans have decoded our human genome, which means they have figured out all the base pairs that make us up as a human to make a human, not like a rabbit or a cat, just a human, which is very incredible, and it's a step in advancement in our life to make our life better, as we can make a lot of new discoveries with that. And this is basically the same thing here, except what happens is thymine is not there present in the RNA. It's replaced with uracil. Well, these are all types of chemicals, by the way. Uracil pairs with adenine in the case instead of thymine, and cytosine pairs with guanine as well. Not really pairs, like they're not really pairing up. They're just sequencing. They're sequenced like that. So there are types of RNA. There are very many types of RNA, which I will talk about. There's a messenger RNA, which is like a RNA. It's like a think of it like a moving. It's like a walking photograph. Basically, it can move and it records things inside of it. And to think about it, anything that records things is pretty it's very important because as you know, if computers, their main job is to record things and data like in bit forms of ones and zeros. If you're using a computer, any a digital like that, they have to store it. And if anything you want to store anything, that's like a powerful thing. So mRNA is very useful in DNA replication and protein synthesis, which I'll be talking about later. There's ribosomal RNA. Ribosomes are the things that make proteins, and proteins are basically what they do is they just they dictate, well, what color of skin, what color like eye you have, what what are the traits that you get, and basically they make you as a whole. They're like the building blocks. So that's why people say eat your protein. Like if you're if you eat meat, meat has a good source of protein. If you don't eat meat, there are a lot of other things with protein. Like I've heard soybeans have protein, that like they're rich in protein. So proteins are very important in this world in microbiology, as you know. That's in every single meal you have to have protein in your meal. Yeah. This is tRNA, transfer RNA, which basically means it's like a. I'll talk about it later. It's if it looks like a weird shape, it is. It has an amino acid at the end, which is basic. Proteins are all made of amino acids, so I guess you can guess about what this tRNA does. It builds the proteins. So this is DNA replication. DNA replication is pretty. It's very interesting in like how your how your body does this, because firstly you have a this splitting mechanism. So you have a topoisomerase which basically unwinds the DNA, and if your body will tell your nuclear whenever the time it is to make proteins, or replicate DNA when this cell is multiplying. Um the your your cell will automatically do this. It's very fascinating how it does. Basically, it unwinds the DNA because the DNA is all coiled up in that double helix form. And then after that, it goes to a splitting phase where you split both of the both sides, so the original DNA, and then you have like the other side of the original DNA. So basically, now you have a DNA polymerase, which is like a scanner almost. It's not really. It's like basically what happens is since you know that si- uh, since you know that thymine pairs to adenine and cytosine pairs up with guanine, you know that they will both automatically replicate the DNA. So that's ingenious, because if you see here, you only have one pair of DNA, but you can use it like a puzzle. You fit the puzzle pieces, because cytosine and guanine have to go each other. They're chemically like that, and thymine and adenine also have to go together. So the the RNA pieces will automatically click in. The DNA pieces will also automatically click in, and it's like a replication. It's very ingenious. And then after that, this DNA polymerase, makes sure they're secure and winds them back up into new the new DNA. Yep. So whenever your body needs to make a protein synthesis, it basically does this step first, and then what happens is it sends out the messenger RNA out. Basically, you see this. Instead of rewinding it back, it only rewinds like part of the DNA, which is the original DNA. It rewinds that up, and then it has the leading strand. It sends only half of the strand. Basically, it doesn't pair it up with its part. It sends it, and that's called mRNA. So the mRNA basically leaves the nucleus, goes to the cytoplasm, and what happens is it goes to a ribosome. Ribosomes are these like really tiny. They're like, if you look from outside, you would think they're useless, but they're very important in life, and because they make the proteins, and your body has tons of these because your body always is constantly making proteins for you to function. And because proteins are basically like almost they are the basic building block apart from cells. They're the ones behind the cells. Behind the proteins are amino acids, which are incredible. To think that we come from, something like amino acids is crazy. So what happens is the mRNA comes up and it reads each base pair into three. So it would be like CTC, like CTC. So C pairs with G, because cytosine guanine, and then T pairs with A, and then that would be adenine, and then C, another C pairs to G. So if you don't get this, basically it reads it into three, and each and every three pieces that you have, for example, the transfer RNA, each and every three pieces, it has one transfer RNA for it, and that has a certain amino acid in the top. And basically, it keeps on doing that until it keeps reading 3 by 3 base pairs, bases, and after that, you call each of these a codon, and these transfer RNA has anticodons, which basically pair up, because as you know, it kind of reverse engineers its way to what the DNA is trying to say, and basically you now have a codon with the opposite of what you want in the code. You have an anticodon which is opposite of the codon, so then that has a amino acid sticking to the end, and basically as you have more anticodons pairing up, you have all these amino acids which basically grow up in a chain. The tRNA leaves. There's only three tRNAs are allowed. So one is ready, waiting to bind on, one is reading it, and the other one's leaving. That's basically how it works, and it's called a translation. And then now you see a newly formed protein here. It's very ingenious. And how there are different types of proteins. For example, um, you have to read the first letter. First, if the first letter is C for cytosine, okay, then you go to the second letter. Second letter is U for uracil, because obviously DNA does not. This is messenger RNA, and RNA has uracil instead of thymine, so it looks for uracil. So CU, and then it looks for the last one. So for example, if it was U again, it would be histamine, which is which is an amino acid, and basically you can read this table chart, and there's a stop, there's a start here, TRP, start, there's a start protein, which means, it's the stop protein means, just terminate this protein. And basically what happens is the protein folds into a way. It folds in, and like kind of like this, it folds up, and they are amino acids are chained by peptide bonds, and that's how they chain up. And then the way the protein folds up is what dictates what jobs it can do. So there are different characteristics of each of these amino acids, and when you combine them all together, this protein can fold up. Where do you get these proteins from? These proteins are from the things that you eat. So that's why it's very important for you to eat proteins, so that your body can make new proteins for you to be useful for. So that is the video. I hope you learned a lot about this genetic material, DNA and RNA. It's honestly a very ingenious way how our body works, and if you want to learn more, there are many more videos that I will be posting on my channel, if you can see, and hopefully you can see them and reflect on um how how amazing nature is, and how amazing biology is for us. All right, that's it for today, and I'll see you next video.